Good morning to everyone. Today we are going to discuss about the topic on introduction to the gene transfer in microbes. I am Dr. Krishna Kumar, Head and Assistant Professor, Department of Botany, Cardamom Planter Association College, Bodhinayakanur. Let's move on to the topic. Bacteria reproduce by the process of binary fusion. In this process, chromosome in the mother cell is replicated and copy is allocated to each of the daughter cells. As a result, two daughter cells are genetically identical. In certain events, change the bacterial chromosome and then these changes are passed on to the future generation by the binary fusion. In this chapter, we will explore some of the events that result in heritable changes in the genomes, gene transfer and recombination, plasmid and transposons. What do you mean by recombination? The genetic recombination is refers to the exchange to DNA molecule. It results the new combination of the genes on the chromosome. The most familiar with a recombination event known as crossing over. In crossing over, two homologous chromosomes breaking at corresponding point switch, shift and rejoin. This result, two recombination chromosomes are formed. Gene transfer. Genetic transfer is mechanism by which DNA is transferred from donor to the recipient. Once the donor is inside the recipient, crossing over can also occur. This result is a recombinant cells that has genome different from either donor or recipient. In bacteria, genetic transfer happens in three ways: transformation, transduction, and conjugation. In recombination, events must occur after the transfer in other that changes in the genomes is be heritable. Transformation. What is transformation? After cell lysis or death in the bacterial cells, release the DNA in the environment, other bacteria. Generally, these are same species. Can come into contact with these fragments, take them up and incorporate into their DNA by the recombination. This is called transformation. Next, move on to the conjugation. What do you mean conjugation? The conjugation is a process by which one bacterium transfers genetic material to another through the direct contact. During the conjugation, one bacterium serves as a donor of the genetic material and other serves as a recipient. Donor bacterium carries the DNA sequence called fertility factor or F factors. Next, move on to the topic transduction. Another method of genetic transfer is recombination is transduction. In this method, involve the transfer of DNA from bacterium to other bacterium use of bacteriophage. Last topic, transposon. What do you mean transposon? Transposable genetic elements is called transposable genetic elements. Or piece of DNA can move from one location on the chromosome to other, from plasmid to the chromosome, or vice versa, from other one plasmid to the other plasmid. That is called transposons. That's all about that introduction about the gene transfer in bacteria. Thank you.